Welcome to the Explainer. Today we're shrinking down to take a microscopic factory tour of the human cell to see exactly how it operates. Imagine a manufacturing plant so advanced, so flawlessly automated, and honestly so intricate that it makes our most modern human-made factories look like simple clockwork. Well, that's the biological reality happening inside you right this very second.
Okay, let's quickly check out our tour itinerary. First, the microscopic factory tour itself. Then, we'll walk the endomembrane assembly line, check out the fluid mosaic model, dive into phospholipids as a moving sea, look at proteins and cholesterol, and finally, review the final blueprint.
All right, Section 1, the microscopic factory tour. Before we look at the machinery, we really have to understand the building itself. Ever since Robert Hooke first observed a quark sample and saw those tiny, empty box structures he decided to call cells, our understanding has completely revolutionized. Thanks to scientists like Schleiden, Schwann, and Virchow, we now operate on cell theory. It breaks down into three simple things. First, all organisms are composed of cells. Second, the cell is the absolute basic unit of structure and function, literally the smallest unit capable of conducting all metabolic activities. And third, and this is absolutely crucial, every single cell comes from a pre-existing cell through self-reproduction. It is the ultimate self-replicating machine. Moving on to Section 2, the endomembrane assembly line. Now, notice how clearly our manufacturing center is divided. Over here is the endoplasmic reticulum, or the ER. It's essentially this massive network of membranous tubules and sacs. That section studded with all those little dots, the ribosomes, is the rough ER. This is your protein synthesis department, creating proteins that get released directly into the lumen. Right next to it is the smooth ER, which entirely lacks ribosomes. This is the lipid manufacturer, synthesizing fats and modifying them. This entire system is just a flawless workflow. First, the products are built in the ER. Next, they get placed into tiny membranous spheres called transport vesicles. You can kind of think of these as our cellular forklifts. These vesicles bud off from the ER, carrying glycoproteins or lipids, and are physically transported through the cell to our next stop for modification before they're finally packaged up for their ultimate destination. And this brilliantly illustrates the Golgi apparatus, which is our factory's shipping and receiving department. Notice how it has a distinct directionality. It takes in those transport vesicles we just talked about at its cis face, that's the receiving side. The vesicles fuse with the Golgi, emptying their contents inside. Now, as these proteins and lipids travel through the Golgi towards the transface, the shipping side, they undergo further vital modifications. Finally, they're sorted, packaged into brand new secretory vesicles, and dispatched to other locations or sent out of the cell entirely.
Okay, Section 3, the fluid mosaic model.
So the crucial point here is that the factory well isn't made of rigid bricks. It's actually a living, shifting sea. This concept is captured perfectly in the fluid mosaic model proposed by Singer and Nicholson back in 1972. The fluid part means the core structural components can actually move laterally, side to side along the membrane. The mosaic part refers to the incredible variety of globular proteins embedded within this shifting bilayer, which creates a dynamic, speckled appearance when you look at it from above. Let's take a cutaway view. This selective barrier separates the bustling interior of the cell, the cytoplasm, from the outside world. But it doesn't just lock everything out. It exhibits selective permeability. This means it allows specific, sufficient passages of oxygen, nutrients, and waste products to cross it more easily than other molecules. It's basically a highly intelligent security checkpoint.
Which brings us to Section 4, phospholipids, the moving sea.
The foundational unit here is the phospholipid. Look at how they arrange themselves in a bilayer. They have hydrophilic or water-loving heads that point outward toward the watery environment outside the cell and the watery cytoplasm inside. Meanwhile, their hydrophobic or water-fearing tails point inward, completely hiding from the water, creating a secure, nonpolar interior core. This spontaneous arrangement is exactly what forms the continuous boundary of every single cell. Let's see how this structure builds into that fluid nature we discussed. Notice how some of the hydrocarbon tails have little kinks or bends in them due to double bonds? This is a genius design. If all the tails were perfectly straight, saturated fats, they'd pack together tightly and the membrane would become highly viscous, kind of like cold butter. But those unsaturated kink tails physically push against each other, preventing the molecules from packing too tightly. This keeps the membrane beautifully fluid, allowing that lateral movement across the surface. Moving to Section 5, proteins and cholesterol. The mosaic part of our model is made up of proteins, specifically integral or intrinsic proteins. These act as massive gates. Because they span across the membrane, they're amphipathic, meaning they have a dual nature. They feature nonpolar hydrophobic middle regions that lock them perfectly into the membrane's lipid core. Meanwhile, their polar hydrophilic ends stick out on either side, perfectly designed to interact with the aqueous environment inside and outside the cell. Now, these protein operators don't just stand there looking pretty. They are the true workforce of the cell membrane, performing 6 major vital tasks. They act as transport proteins, allowing specific molecules through. They conduct enzymatic activity to speed up chemical reactions. They handle signal transduction to pass messages into the cell. They allow for cell-to-cell recognition, often using attached carbohydrates. And they serve as attachment points, joining cells together or anchoring the cell's internal skeleton. The diversity of function here is just incredible. Take a look at the transport protein. It might provide a simple hydrophilic channel for polar ions to slip through the hydrophobic wall, or it might literally burn cellular energy, ATP, to actively pump molecules across the border against the normal flow. From sending complex chemical signals down the line to acting like an enzymatic assembly line right on the cell surface, these proteins absolutely make life possible. And surprise, cholesterol isn't just the bad guy on a nutrition label. In animal cells, cholesterol molecules are tucked right in between the phospholipids. It actually acts as the essential thermostat of the membrane, stabilizing its structure. When temperatures get dangerously warm, cholesterol restricts the movement of phospholipids, making the membrane less fluid, so it doesn't just melt apart. Conversely, at low temperatures, it wedges between the phospholipids to avoid close packing, preventing the membrane from freezing solid. It's the ultimate temperature buffer. Finally, Section 6, the final blueprints. As we zoom back out from this microscopic factory, leaving the dynamic fluid borders, the transport vesicles acting as forklifts, and the sprawling assembly lines of the ER and Golgi, it is truly breathtaking to realize the scale and efficiency of this biological engineering. When we talk about engineering marvels, we usually look to skyscrapers or supercomputers, but the true masterpiece of automation is the eukaryotic cell. I'll give you with this provocative thought to ponder. How many billions of these flawless microscopic assembly lines are running perfectly inside you at this very second, coordinating flawlessly without a single conscious thought from you? Thanks for taking this explainer tour with me, and keep exploring the incredible microscopic world inside you.